All right, welcome everybody. Tony had a conversation, I guess, with Chad at one of his open houses, and the next time I saw Tony, he said, you have got to have this person here to give a talk. This was fascinating stuff. He told me a little bit about it, and I went home and said something in front of my daughter, and she said, "Oh, great, Dad, you can go on your favorite rant about space plants," which is in The Martian, which is one of my all-time favorite movies. I love that movie. But when he's up, if you've seen it, Matt Damon there, he's in there talking to the camera, and he says he's not only whatever he is, I can't remember, he's also, and he holds up a book, the botanist on the trip. And then he talks about so he can grow plants. And I'm sorry, botanists study dead plants. They don't know how to grow plants. He's doing horticulture, not botany on there. So I will leave it at that. Chad is a research scientist at Redwire Space, where he develops biology payloads and supports a variety of science investigations through the International Space Station. Works closely with NASA and the international community of researchers to grow and study plants in microgravity. Chad also serves as a plant physiologist with the Space Agriculture Laboratory Analysis Database, or SALAD team. I swear to come up with a word and say we can fit it all in there. What starts with S for space? Chad earned his master's in space biology and plant physiology from the University of Louisiana at Lafayette, where he worked in Carl Hastenstein's lab. He has a BS in horticulture, so he's doing real plant stuff here. From Michigan State, hold your applause. And I just found out tonight that he works with, he did work with Tom Ranney, got to know Tom Ranney when he worked at Spring Meadow and worked at Walter's Gardens with Hans Hansen. When I heard that he worked with Hans and knew Tom, I knew he was the real deal. The space stuff, whatever. But anybody who hangs out with them knows what they're doing. So we're going to talk about astrobotany, perhaps astrohorticulture too, 101, earthly roots, cosmic ambition. So please help me welcome Chad Vanden Bosch.
All right, well, thank you. Can everyone hear me okay? Is the mic picking up my voice? Wonderful. All right, well, thank you, Mark, and thank you all for being here. I'm excited to be here. I've been here to the Arboretum a few times now, and the grounds are just lovely. In fact, I came here on Monday with my mother, and we toured the grounds for a few hours and showed her some of the beautiful plants that we have growing down here, and she really, really enjoyed it. So tonight we're going to talk about astrobotany, or as Mark said, it should be called astrohorticulture, perhaps. And we're just going to get into it. But before we talk about astrobotany, I'm going to tell you about myself a little bit. So I grew up in Michigan. I was born and raised near Holland, Michigan, which is close to Grand Rapids on the west side of the state, where that star is on the upper left. And I always had an interest in plants. My mom always had a garden, and I always helped her in the garden. I would pick tomato worms for 25 cents a piece, and that was decent money back then. But back in 7th grade, I actually started working in a greenhouse after school, and I really loved it. I enjoyed it. So I worked in that greenhouse from 7th grade all the way on to my sophomore year of community college. And I decided to transfer to Michigan State, and I realized, I really love horticulture. I love growing plants. May as well try and make a career out of it. So at Michigan State, I got a bachelor's in horticulture. And then I moved back to the west side of the state. And it was there that I got to work for Hans Hansen at Walter's Gardens. I worked with him for just over a year in their plant hybridization department. So I was kind of helping them with different cross-pollinations. I was kind of their interim grower, so I was doing a lot of the watering and plant propagation, and some selections as well. So that was a great introduction into what the world of horticulture can really, really hold. And you guys have probably heard Hans speak, it sounds like, a few times, and he really just is a walking encyclopedia of plant knowledge. It's amazing how much he knows and he can share, and he's such a nice and humble person too. So that was a great kind of intro into the field of horticulture. From there, I actually moved and worked at Spring Meadow Nursery for a few years. At Spring Meadow, I was their trials and propagation manager. So with that, I would propagate all of their in-house trial plants and also all of the plants that came from their network of breeders, which is largely in the US, but also there's some global breeders that ship them their varieties of flowering shrubs for trialing. So I would propagate all those plants, usually, ideally at the same time, and then we run trials in containers inside the greenhouse. We do field trials, and we also would conduct trials in a more formal garden-type setting. So through those means, we're able to kind of determine, which plants, A, propagate well, B, perform in a container, C, work well in a garden setting. So that was between Spring Meadow and Walter's Gardens. Those were two really great experiences in not only plant propagation, but horticulture in general. But after a few years, I kind of thought I want to try something more, try something new. What's really novel, and is what's really piquing my interests, and that was space biology. So with that, I kind of reached out to a few different folks that were doing research with NASA, through NASA, and just tried to figure out who may, have a position open in their lab that would take me on. And I was lucky that Dr. Hassenstein, who works for the University of Louisiana, had a position open. So I hopped on board and moved on to Louisiana. And that was in January of 2020. And interesting year for all of us. But amazingly, my girlfriend, now wife, Gabby, also was willing to move down there. That was in the summer of 2020. So things were weird in January and even more strange in the summer. So we did a midsummer move down to hot and steamy Louisiana. And it was a 16-hour road trip drive from Michigan. And that was... it was not so much fun, but we made it. We survived. So in Louisiana, in Lafayette, I studied astrobotany, or more just generally space biology. And at the time, my advisor was working with NASA, and he was growing radishes on the space station. That was his current research project. And with that, he was working with a company that kind of helped to, we'll call it, integrate his science into the ISS. So he had a plan, a proposal. After NASA says, yes, you have a great idea, let's make your vision, your dream into reality, they then took his proposal and passed it off to another company. And this company was Redwire Space. So through my grad school years, I got to know the folks at Redwire Space. And I was very fortunate enough that after I graduated, they had an opening, and I applied, and everything just kind of fell into place. So after Louisiana, we moved to Florida. And we lived in Florida by the Kennedy Space Center for just about 3 years. We have an office, Redwire has an office right on Kennedy Space Center property. So it was... you're at the forefront of the gateway of space. It's a really fun place to be. There's rocket launches every 3 or 4 days now. There's over 100 a year that launch from there. It's just, if you're into space, it's the place to be. It's really cool. Yeah. But after a few years, we wanted to be close to family, and my wife, Gabby, has family in Winston-Salem. So we started kind of looking at North Carolina, and Gabby just happened to find a job with the city of Raleigh, and that brought us up to North Carolina. And that was on New Year's Day is when we moved up here. So we're kind of fresh to the area, but we're loving it thus far. And the gardens are beautiful, and we're having fun exploring the area. So that's a bit about me, just kind of laying some backstory. So what is astrobotany? Of course, we have to talk about the Martian. That's already been discussed, so I won't belabor that point. But essentially, I would define astrobotany as growing plants anywhere but Earth. Typically, at this point, it's growing plants on the ISS or on the Chinese space station, or maybe some sort of free-flying satellite type module. But eventually, hopefully in the very near future, we could have plants that are growing on the moon. Plants have been grown on the moon once before. The Chinese actually sent up a lunar lander and germinated some cotton seedlings. I think they only grew for like 3 or 4 days, but they did sprout and grow. So that's something. So that's kind of my loose definition of astrobotany. And we see this really lovely image on the left of some zinnias that were grown on ISS, I think way back in, sorry, not way back, but back in 2013. So a few years ago. So I'm just going to kind of lay out some of the basic steps to spaceflight, at least from what we do from a perspective at Redwire Space. So I kind of talked about how my advisor had his radish proposal accepted. So that's the first step to any spaceflight is NASA may put out a call or a solicitation for proposals, and it may be based on a certain growth chamber like this one or this one, or it could be for any other number of growth chambers or environments that are available on the space station. So they'll put out a call for proposals. Then often university professors or sometimes private industry will then write in with their great ideas. And then it's kind of NASA's job to narrow them down and select just one or maybe two. At that point, NASA then takes these researchers and their proposals and kind of passes them along to us. So we kind of take their science, their vision, and try and turn it into a reality. One guy wanted to grow, I think he wanted to grow 250 cotton plants on the ISS, which is a great idea, but there's simply not enough space for that, right? So what we did is we asked, well, what are you really trying to study, or what do you want to see? And he's like, well, I just want to see how the cells respond to the space environment, microgravity, radiation, that sort of thing. So we asked him, well, what about tissue culture? Could you tissue culture your cotton plants, and can we fly some cali instead? And that's exactly what we did. So it's a matter of like taking somebody's dream and finding a way to make it work up on the ISS, which typically is very spatially constrained. You're constrained by crew time and many other things, but space and crew time are probably the biggest things. So anyways, we'll kind of define their science, figure out exactly what it is they want to achieve, and what are they looking to study and return from their plants. Then before we do any flights, we'll do some ground studies. So it's obviously pretty expensive to fly anything up out of the Earth's orbit and up to the ISS. So before any of that happens, NASA wants to really be, as sure as possible that this experiment will indeed work up there. So we'll do usually 2 or 3 studies on the ground before we fly anything. So these are two growth chambers here. They're both called the plant habitat, or the advanced plant habitat. But for every single growth chamber, there's usually at least one on the ISS, and there's usually multiples of them on the ground, typically at the Kennedy Space Center. So prior to any flight, we're going to do at least 2 or 3 ground grow outs and just vet their experiment, try and dial in the environmental controls. Maybe it's atmospheric composition, the CO2 levels, the relative humidity levels, the moisture levels, figure out days from germination to harvest or thinning or whatever it may be. Whatever they're trying to study in their proposal, we're going to try and vet that and get that all worked out on the ground before it actually comes time to fly this thing. And all the while, we're also taking a lot of notes and trying to figure out how do we take all this knowledge that we and the researchers have, and how do we put it onto a piece of paper or an iPad and translate it to an astronaut who's probably a former fighter pilot and make them, a stand-in plant scientist. And that can be a real challenge at times, actually. So that's kind of step 4 is we take everything that we need to fly, whether it's the plant seeds, we get the actual we call this, I guess, a facility. We get the facility ready. If they need to have any parts replaced, we'll make sure those parts get flown up prior to our experiment. They get replaced by the crew. So hopefully the facility is all set for us by the time it comes to actually start the experiment and grow these things. We're also working through, up mass, down mass, the volume of everything that we're flying. Everything needs to be recorded and known because space on a rocket is very valuable and very limited. So they want to know as much as possible about the science that we're going to fly before they actually fly it, which all makes sense. So then the exciting part is actually watching a liftoff. And typically those occur from the Kennedy Space Center. There's, over 100 per year. And there's a few ways to get these plants into orbit. Usually it's on a SpaceX Falcon 9 rocket, which is a reusable rocket, and it's really fun to watch because there's two pieces, and the main body of the rocket will kind of split from the topmost part. The topmost part keeps going, and the main body kind of floats and then actually kind of boosts back and lands on the ground. And if you're watching it from the Space Center, you can watch that all unfold right in front of you. And it's just, if you're not like a fan of space, I get it. Not everyone is, but like watching that is pretty fantastic. It's really cool. And then there's the sonic booms that come with the booster as it comes back down too. It's pretty fun. So anyways, that's the really exciting part. That's kind of where we're really making some progress here. It's probably been, from the proposal acceptance to liftoff, it's probably been about 1.5 years or 2 years if everything works smoothly and works well. So it takes a while. And then once we're actually on the space station, the crew would then, unpack everything that we need and hopefully install the science. We could start the growth cycle. So on the bottom right here, we see 2 crew members. This is Sunny Williams She was just on ISS for 9 months. She was on maybe you've heard, but she was on the Boeing Starliner, which is kind of infamous. It was supposed to be a 9-day tour, but it was a 9-month tour, kind of Gilligan's Island-esque. But she survived and she made it, and she was great and really, really fun to work with. And you can see, they're very happy to be working with what were plants in that growth chamber. They just harvested the plants. We grew some lettuce up there. So while the crew is kind of working with the plants, whether it's thinning or harvesting, whatever it may be, we're on console, we call it, and we'll have like a headset on, which you can see me wearing there, and we'll have like a direct communication with the crew. So we'll be able to watch them on a video camera, and if they're kind of having trouble with something, we can call up to them and say, "Hey, Sunny, is everything okay? Do you need any help?" And she says, "No, I'm fine." Then we just let her work. If she has a question, we'll try and help her out and make sure everyone's happy and we get a good return on the science. So while that's happening on the ISS, we're also in parallel, we're conducting ground studies. So we're going to do the best we can to replicate what happens on station on the ground. So our ground is kind of a comparison or a basis for comparison from flight to ground. So we can't emulate, the forces that are created by the rocket going up or returning back to Earth. We don't emulate the radiation, thankfully. And we also can't really emulate microgravity. But everything else we try to, whether it's the CO2, which on the ISS, it's anywhere between 3 and up to 8,000 parts per million. So it's really elevated. This growth chamber can actually control CO2. It can scrub it down. So it could go as low as 1,000 parts per million if you wanted to. It all just depends on what is best for the science and for the plants that are being grown in whatever facility it is that we're using. But we'll try and match the CO2, the relative humidity, and the moisture environments. Then of course the timing of the experiment as well. If we're growing the plants on day 0 and by day 8 they need to be thinned, we're going to try and do our thinning on day 8 on the ground as well. So we'll just try and match flight as best as we can on the ground to get a good control for the flight experiment. And then once that all happens, we need to get the plant specimens back to the Earth. if the crew haven't had a few nibbles or eaten all of them, which happens sometimes. But this is really, the most important part, because we can do all these steps 1 through 7, but if we don't get the samples back to Earth, then there really isn't a whole lot of use, at least for us or the scientists who kind of, have been working for 3 or 4 years to try and get this all to work out for them. So often we'll freeze things. They have freezers up on the ISS that can control down to -20 C or -80 C. So we'll kind of deep freeze things, or some things can come back refrigerated. It just depends. But most things come back frozen, and some can come back in like a chemical fixative too. There's different methods that we can use. But this is just kind of like the groundwork to what I do at work every day. And if you have any questions along the way, please just chime in, raise your hand. I'd be more than happy to pause and answer any questions. So I don't know if I'm going to fill a full hour, but I'll try to. I won't try and be too long-winded, but any questions, please just shout them out. Okay, so I kind of divide astrobotany or astrohorticulture into four main tiers. The first is physiology. So how do these plants actually function and respond to this really unique environment? Plants evolved to be, an organism here on Earth with gravity and a certain atmospheric composition. So how do they change and respond when there is almost an entire lack of gravity on the ISS or other platforms? So that's kind of the physiology aspect. Of course, there's more than that, but mainly, when you're talking about plant physiology research on ISS, it's mostly lack of gravity, high radiation studies, and a lot of researchers look into omics. So maybe it's genomics or proteomics. How are they responding, maybe instantaneously to this new environment, and whether it's through RNA analysis, it could be more or less a snapshot of that plant at that given time. And if you can preserve, say, a snipped leaf in something like RNA later, which is a plant fixative, you can then figure out exactly how that plant is coding its DNA at that time through RNA. You can figure out how it's responding and functioning at that single instance. And you could take RNA leaf samples. You could take samples, once every day for 2 weeks, and maybe determine how they're growing or responding to whatever stimuli they're receiving at that time. The second tier is crop production, growing food to eventually try and supplement the crew's diets. This is going to be really important, especially if we're talking about trying to go further and further away from Earth. The ISS is a 3 or 4 hour ride from Earth, which is not so far. So they deliver packaged and sealed meals, and those meals last for up to 2 years typically. But over time, those meals and some of the chemicals and the nutrients will kind of break down, especially like vitamin C, vitamin E. The nutrient composition starts to kind of degrade and erode over time. So the thought is, if we're going far away, like Mars, for example, that's probably a 6-month journey year-round. To go back and forth, round trip, it's about a year. And if we're talking a year just in travel time, they're going to need to figure out how to grow and produce their own food at that point. So that's where crop production is really kind of coming into play, and that's where its importance, its relevance will really shine. On the ISS, there's some crop production research. It's right now, it's kind of a blend of physiology and crop production. So we're still trying to really just lay the groundwork and figure out how these plants are functioning and responding to this really strange and unique environment. And I should mention, physiology can inform how plant breeders could actually breed plants, select them for space environments as well. A lot of plants are actually, they experience a lot of like heat and drought stress on the ISS, even if it's normal room temperatures and they're well watered. For some reason, there's something about being in a lack of gravity that causes them to think that they're in this it's a really hot or very dry environment. So maybe, by using some of those genomics, plant breeders could then breed plants that are possibly, maybe they downregulate their drought stressors or their heat stressors, so they don't respond this way when they're being grown on the ISS or maybe some other space station, maybe the Moon or Mars. The next here is bioregenerative life support systems. So on the ISS, there are a number of systems that scrub CO2 from the ISS and eventually make oxygen, which the crew needs to breathe, of course. And they also have like a water recycling system. You've probably heard, but the crew has to recycle every drop of water, and it all goes back to drinking water. So I've heard the water doesn't taste great up there, and I can imagine. But if we can use plants to do that, that's a more natural way. And I'd much rather drink water from that's collected from a plant than from like a urine processor, right? So this is it's really nascent. It's fairly new because we're 3 or 4 hours away from Earth. And we don't have a ton of space to devote to plant research. But this is something that's kind of emerging and will definitely be more important, again, as we go further away, further out from Earth, or if we try and establish, say, like a long-term settlement or a permanent settlement on the Moon or maybe Mars. And lastly, this was kind of in the back of people's minds for a while. But you all know this. I mean, plants really make people happy. And especially when you're in, an ISS, which is just full of wires and cables, and every surface around you from floor to ceiling to the walls are just covered in stuff. I mean, it looks like a rat's nest to me. But they really enjoy working with the plants that we send up there. So it makes them happy. And I think, again, if we're going to try and ever move off planet and become a multiplanetary species, which is an interesting idea, I don't know that we have to do it, but I think it's interesting. But if we ever do, make that leap, we will need to bring our plants with us. And it's not just for nutrition, but also just to keep people happy and hopefully sane as they're away from Earth. All right, so I'm going to delve into some of the past research a little bit, and then I'll try and get to some more modern stuff, and then look into the future a little bit as well. I'll try my best at least. So I would say the first Astrobotany experiment occurred back in 1946, and this was actually on a German V2 rocket. And most of you probably know that actually our rocket program was kind of birthed through a lot of former German scientists who were brought over post-World War II. And they then went on to build the Atlas V, which brought us to the Moon. But after the war, we brought their scientists over and also some of their V2 rockets. And so they decided to launch corn seeds on a V2 rocket, and then they returned them, collected them, and they grew. So that was a good sign. Space didn't absolutely kill these seeds, in the 2 minutes that they were up there. So that's a positive sign. We can move forward. The next step was in 1966, a satellite called the biosatellite, which is this guy up here. It carried wheat seeds into orbit, and this was a longer term orbit. It was up there for a few weeks, and then it had returned. And again, these wheat seeds were not really affected. They germinated just fine. So that's another positive sign. Very good. So on to 1973, with a few of the rockets that we had left over from the Apollo program. These are these big Atlas V, the big 300-foot tall behemoths. There were some left over after the Apollo program was canceled. So they decided to build and fly something called Skylab, which was the first space station that orbited Earth. And me being of Dutch roots, I think it looks like a windmill myself. But on Skylab, they actually decided to grow and germinate some plants. So they grew wheat and rice, and they all survived and grew pretty well. That's another successful sign that plants don't only grow on Earth, but they can maybe survive in space. And in 1983, we had our first Astrobotany experiment on a space shuttle. And this one was actually sponsored by a company who, I believe, is Park Seed of South Carolina. They're from South Carolina? Okay, so a semi-local company. Park Seed decided to fund and fly 25 pounds worth of seeds. And I think they were kind of like common crops, a lot of vegetables and floral seeds. So they flew 25 pounds worth of these seeds up on the shuttle. And I think the shuttle was up there for about 2 weeks. And Park Seed just simply wanted to see, of all these seeds that we fly up there, how is microgravity and the more harsh radiation environment going to influence these seeds and their subsequent germination upon their return. And they found that it didn't really make a difference. They had good germination of the seeds that had flown compared to those that stayed in the ground. There really wasn't much of a difference. So a 2-week stay being exposed to microgravity and this more intense radiation didn't really make a difference, at least not from a plant germination perspective. They seemed to grow just fine. Any questions or comments? I'm gonna grab a sip of my tea really quick. Okay, so what are we currently doing? So there's a few vehicles that are in use for current astrobotany experiments. So this thing looks oddly like a space shuttle, and it's very similar design. It's just quite a bit smaller. This thing is called the Boeing X-37, and typically it's not many people know about it because it's kind of like a secret government-sponsored space plane. But what it does is they'll fly it once every 4 or 5 years, and it's up there for 900 to 1,000 days at a time, just orbiting. And what they're looking at, what they're using it for, I don't know exactly. But what I do know is that NASA scientists have put plant seeds on that thing as well, and they want to see how more of a long-term exposure to mostly radiation would influence plant seeds and their progeny. We don't have the results from that experiment yet because I think this most recent X-39 flight came back last year, and NASA just got the seeds, and it takes a few years to analyze and process them. So that's one way that we're conducting astrobotany today, kind of a secretive way, but work is being done there. Another much more public way is through the Artemis missions. Artemis 1 launched a couple of years ago. Artemis 2 is scheduled to launch next year. But on Artemis 1, they carried plant seeds. They carried Arabidopsis seeds to the moon and back. And you're probably aware, but for those of you that don't know, Arabidopsis is kind of like the model organism of the plant world. You can think of it as like the lab rat of plants. It's very well understood. It has a compact size, a fairly short life cycle, and we know a lot about the genome of Arabidopsis. So it's a great candidate for space because of its size, short lifespan, and our broad knowledge base of that plant to begin with. So if we're thinking about genomics and how do these plants respond physiologically, that's a great plant because we can then decipher which genes are activated or promoted up or promoted down based on the space environment. But the real workhorse is the ISS. That's where almost all astrobotany research occurs today, and the ISS has been continuously occupied it'll be 25 years in November. And there's typically a crew of 8 to 12 these days. So there's a lot of manpower or person power on the ISS, and this whole thing is about the size of a football field. But the actual living quarters, it's about the size of a 6-bedroom house, they say. I'll take their word for it. So there's a lot of space on this thing, and we can certainly fill it up and use it for plant research. I wish more of it were devoted to plant research, but we'll take what we can get. So one such system that is on the ISS that grows plants is called the veggie system, or the vegetable production system. It's a fairly simple payload, and it's mostly used for crop production research. Not too many bells and whistles, but what it does have is it has kind of like an accordion-like greenhouse that wraps around the plants, and it has like a lower deck that can be adjusted and moved up and down as the plants grow and grow up and reach towards the light, which is something else that it has. It has a controllable LED light bank. So they can, I think it's red, blue, green, and probably white. I think white also. So they can tune the LEDs based on whatever the requirements are for their plant species that they're growing. Here we have, I think this is Mizuna. I think this is a red romaine lettuce, and these are zinnias. And that's about it for the veggie system. It's pretty simple, nothing too fancy, but it works, and that's what matters. Plants are often grown in what we call plant pillows, and that's this thing right here. So it's kind of like it's about the size of maybe like 2 fists put together. Think of like the length of a hot dog bun, but the width of a hamburger bun, we'll say. It's not huge, but it's this sewn together pouch, and the pouch is full of a material called arsenite, which is like a granulated clay. And it's actually what they use on baseball diamond running paths, if you believe it or not. That's what they found works best for plants in space. So they use plant pillows to sow the seeds, and they'll fly these plant pillows pre-packed with the arsenite, with fertilizer, and with the seeds already planted onto the wicks. One thing that I've mentioned that's really important to keep in mind is crew time. So we don't really want the crew to have to do any more work than they absolutely have to. So if we can pre-sow the seeds, if we can send them something that's tailor-made, ready to go, all they have to do is unpack it and stick it down to this grid and add some water. That's all the better for them. It makes their lives much easier, and it lowers their demand. And with that, they get to kind of eat the fruits of their labor. So on the left here, these two guys, Scott Kelly and Kel Lundgren, they grew red romaine lettuce. And now they get to eat it, and they loved it. They're super happy about it. Scott Kelly is kind of famous because he did a year in space, and he's also a twin. His brother is now he's a former NASA astronaut. His brother is I think he's a senator for Arizona, correct? Yeah, but with his year in space, they were able to kind of measure his own body responses to the space environment over a year and compare it with his brother's, who was on Earth for a year. So it was a really novel and interesting human physiology experiment that he was conducting as he was conducting this plant experiment. This is another astronaut who's eating, I think this is Mizuna, probably the Mizuna that we saw on this last slide. She had harvested it, and now she's able to consume it. So fresh food is something that doesn't fly up to ISS very often. They don't have a whole lot of fresh food. It's mostly prepackaged stuff, and they throw it in some sort of like a warmer or like a microwave type oven, and they eat it out of this package. It's something like NASA space age Lunchables or something. Supposedly it tastes good, but I don't know. There's nothing like, a fresh salad, right? And we can have that every day, all day if we want to, but they don't get to have this stuff hardly ever. So when they do get to eat it, especially when they're growing it themselves, they're really excited about it and they enjoy it. And that's the psychological aspect that we don't often think about when it comes to growing with and working with plants, is they really get a lot of joy out of that. There's another item called the X-roots. So X-roots uses this same growth chamber. It uses the veggie growth chamber, but in a different system. It doesn't use a plant pillow, but instead it uses kind of like a plastic 3D printed container. And instead of using like a media to grow the roots in, they actually just let these roots hang out in this container, and they shoot them with a mist periodically. So it's an aeroponic system. And that has worked really well. I think we have radishes that are shown here, but they've grown radishes, tomatoes, and lettuce in this system. And it's a really nice proof of concept in that you don't need to launch these plant pillows, which take up space, volume, mass. They're kind of heavy. And the crew has to manually water these plant pillows, almost every single day when these plants are growing, and when they're larger, like this size, they're probably watering those plants every single day. And that, again, is they may like doing it. I would like doing it, but it's a demand on their time every single day. So if we can automate plant growth and just use a mister that's on like a pre-programmed cycle and kind of engineer the human out of human spaceflight, we like to say, then that is all the better. And the plants seem to grow and respond quite well. This is kind of bright, sorry. So one system that I worked with is the advanced plant habitat. And as I mentioned before, this is kind of more of a high-tech growth chamber. So the veggie is pretty simple. It has lights, it has a movable greenhouse type covering, and it does have a fan as well. This thing has about 200 different sensors in it. So it can control CO2, can control relative humidity, the moisture environment. So we'll call this is a root tray. And this whole thing is removable, but it plugs into a power bank in the back here. And these 4 quadrants, you see there's 4 separate compartments here. We control water into each one of those 4 quadrants. So recently we did an experiment with NASA where we grew lettuce up in the ISS, and they wanted to study how does variable water affect lettuce from a nutritional aspect, but also from like a microbial aspect. Does a lettuce plant that is really drought stressed, does it have more microbes that aren't great for eating? Or is a plant that's been nearly flooded for all of its duration, its life on space, on ISS, does that plant have maybe any microbes that would upset our digestive system? That sort of thinking. So with that, we grew lettuce plants in these 4 quadrants, but kept all 4 quadrants in different moisture environments to try and pull out whether or not there are any microbial differences from different water stresses. These are radishes, as you can see, and these were 28-day-old plants. So they were very, very happy, and they grew. I mean, they almost filled the entire volume of this chamber. And the chamber is about the size of like a microwave oven. It's not very big. So to plant 20 radishes within a microwave oven, like, we always knew it was going to be tight. But we didn't expect this. I mean, they really grew quite well, and you can see some radish bulbs or hypocotyls really performed and were quite large. On the right-hand side, we have chili peppers. And this is one of the longest experiments that we've run. These were grown for about 170, 180 days. And this was a really interesting proof of concept in that we determined and proved that we could grow fruiting crops and plants in microgravity. All that we asked of the crew was to we're really concerned about pollination because there's not a lot of, there's some airflow in this growth chamber, but there's obviously no bees or insects to move the pollen around from one flower, from one plant to the next. And even if chili peppers are self-pollinating, it's always helpful to have pollen from another plant to really encourage fruiting and subsequent seed development. So we had the crew go in actually with paintbrushes and just kind of brush the flowers every so often, and I think they enjoyed that. And it really worked. We had a good crop. You can see on the bottom here, they harvested a series of red, and I believe he has some green peppers in that bag, which you can't see very well. But it was very successful. And with that, they had a taco night. They had a taco party. Who doesn't like tacos, especially when you can grow the lettuce or grow the lettuce and the peppers yourself and make tacos out of it. So with this growth chamber, we can do both crop production type work and plant physiology work. Because as I mentioned, we have a ground unit at the Kennedy Space Center. So all the while this took place on orbit, we were doing the exact same work on the ground, trying to mirror what the astronauts, what the crew were doing. We were mirroring that on the ground. So we actually put the entire growth chamber in an even larger growth chamber. So it's a bit like Russian nesting dolls. We walk into a big walk-in growth chamber that has our growth chamber within it. But the big walk-in growth chamber is emulating the ISS atmospheric conditions. So ISS is typically around 40% relative humidity, with CO2 ranging between 3 to 5 to 8,000 parts per million. So that's what the big walk-in chamber is programmed to do. It's using ISS playback data to mirror those environments. And then, of course, we're using our growth chamber on the ground to mirror what's happening in this growth chamber on the space station itself. So we typically run a ground control that is anywhere from like two days to a week after the fact. So we can have time to respond to any anomalies, say a CO2 pump or a CO2 filter breaks, and we have high CO2 in the growth chamber for a week. We need to respond to that and be able to replicate that on the ground, because we don't want to make extra variables from the space to the ground study. So we try and mirror space as best we can on the ground. There's taco night. Okay, so we're going to talk about bioregenerative life support really quickly. So this is an artist rendering of what bioregenerative life support should and maybe could look like, somewhere in the near, or probably more likely the far future. So essentially, again, we're using plants to clean the air, to scrub CO2, create oxygen, and hopefully also help to recycle some water, through evapotranspiration. They're taking in water through their roots, they're releasing it through their leaves, and that water is pretty clean. So we're using plants kind of in an integrated system along with some mechanical systems to create a livable, breathable environment for humans. This is what it really looks like. So it's a pretty stark difference, but you got to start somewhere. So on ISS, there's not a whole lot of energy or time that's devoted to these bioregenerative systems. But we're starting to work on that because we're trying to eventually go to the moon, and eventually Mars is the thought. So this is a pretty modest and simple experiment. These are simply like animal cell culture bags that we used, and we filled them with 3 different species of algae. So we were working with a team out of India, and their lab is an algae lab, and they study edible algae. And they also breed algae that is with different or varying concentrations of different phytonutrients. So they wanted to test their algae species, their selections on the ISS, to kind of vet them and see how they responded to microgravity, the radiation, and simply see how they grew in an ambient space-like condition. So these were just Velcroed to a wall, actually, and they grew on a wall for 12 days on the ISS. And when it came time to actually harvest them, we took a sample out of each bag. We put it in like a small vial and we centrifuged it. We made a pellet of that algae. Then the crew came in and very delicately they pipetted the supernatant, the water, out of the vial and left just the algae pellet, which we then froze and then brought home and then shipped that back to India. So very, very simple experiment, but there's some real artistry and technique to it. And we were fortunate that the crew was very, very competent. They had training in pipetting, thankfully, and it worked really well. So this happened just a few months ago, actually, and we're still waiting to hear back from the Indian researcher team to see how it all went, how their samples responded. Yeah, question?
How did they use the water that was left from those pellets?
How did they use it? Oh yeah. So sure, yeah. So the question was, how did they use the water that they decanted from the pellets? So they simply they took the water out with a pipette, and with that pipette they squirted it into a bag that had a cloth in it, and with that they eventually they ended up trashing that water, actually. But if they have water that's like cleanish, we'll say cleanish, they can take like wet cloths and just leave them out to dry, or put them in front of a vent, and then the water reclamation system will pull that water out of the air. Then they can then recycle it. But because these algae species were kind of an unknown for like flight safety perspective, they didn't know how they may impact the crew's bodies. They just decided to throw away that little bit of water that they generated. And it actually, we flew these bags already like pre-filled with water and a nutrient solution. So it wasn't like a net loss of their water supply, which is something we have to be very like cognizant of. It would have been nice if they could have simply added this to their supply, but it's not worth the risk. Yeah, great question. Thank you. So this other system has a little bit more going on. So this is something that the DLR, or the German Space Agency, built and is actually running on ISS right now. So this is an algae photobioreactor. So we've got a bunch of algae here in these tubes, and there's water and air that's being pushed through those tubes. And all the while, there's an LED light that's running behind those tubes, so the algae are able to photosynthesize, and in the midst of that, grow, reproduce, expand. And to do that, they're pulling nutrients out of the water. They're using using CO2, creating oxygen. They're using oxygen during the night cycle, but when the LED is on and running, the algae are growing and they're pulling CO2 out, creating oxygen, and they're cleaning the water. And this isn't like fully integrated. It's not actually plugged into any of like the ISS life support systems, but it's just a proof of concept at this point. Okay. Oh, I see. It's 7:28. All right. I'll try and hurry up a little bit. What time do you guys typically wrap up and have questions? I don't want to talk for too long.
I'm fascinated. So.
Okay. Well, I don't want to put you all to sleep, have an early bedtime, but I'll try and speed it up a little bit and leave some room for questions. But now we're getting into kind of what I think is an interesting part, is the challenges of growing plants in space. So I mentioned that the ISS is kind of a mess. What do you guys think? I mean, I wouldn't want to live in that. I mean, I would, but I wouldn't really want to. So every square inch is covered in cables or laptops or cameras. And, it's been up there for 30 years, and we've been filling it for 30 years, It's like a bedroom that you never clean out. So it's full. So One of our challenges, if we're trying to build, say, a new growth chamber or something like that, is if we build a new growth chamber and we fly it, something else has to come down. Something else has to be removed. They have to decide, like, yeah, it's not worth to keep this system. We're going to pull this system out and replace it with your fancy new plant growth system. That would be great, but it's you're doing some horse trading there, trying to figure out what needs to go in order to make room for what's up and coming. So space is definitely an issue. Crew time is an issue. So I mentioned we try and pre-sow everything that we fly up to the ISS before it's actually up there. So here's a good example. This is a root tray that's going into the plant habitat system. So this root tray is already full of a growing media. It's that arsenite that's on the baseball diamond. That arsenite has slow release fertilizers mixed into it, which once wetted will slowly release their salts and their nutrients. And it may be kind of hard to see, but there's actually a series of wicks on top of that root tray that are pre-sown with seeds all along that wick. So all it took for, his name is Shane, all it took for Shane to do was to unpack this root tray from its bag, slide it into the growth chamber, which is in the process of doing in that image. He hooks up 2 power cables and 4 water lines, then he puts the door back on the outside of the chamber, and then we take control from the ground. We have telemetry, it's all computer controlled, and we kind of run the program. We do manual watering. We do everything that we can from the ground. So we're trying to save them as much time as possible and have everything that is, again, tailor-made, ready and set to go. So he can just take it out of its bag, slide it in, plug it in, close the door, and we're off and running. Another huge challenge is gravity or lack thereof, of course. This is probably more related to growing on ISS. On the moon or Mars, there is fractional gravity. But on ISS or any free-flying spacecraft, there is what we call microgravity. So ISS is always falling down towards Earth, but it's in a trajectory that it's going fast enough that it's always missing and coming back around and trying again. It's perpetual, but not quite. It has to be reboosted every so often to increase its elevation from Earth, because our gravity is dragging it down slowly. So they reboost it from time to time. But that's neither here nor there. But so plants don't have gravity to orient themselves like they do on Earth. So what we use instead is lights. So we're not relying on geotropism or gravitropism, which is gravity movement. We're using light movement instead. So the shoots grow towards the light, the roots tend to grow away from it, or really they grow towards moisture and nutrients, which is why we actually, it's a little tough to see, but this plant root tray has a cover on it, and it's all enclosed, and the wet media is contained. They're very distinct, almost like a membrane or like a cell wall, you could say, between the roots and the shoots. So we're keeping the soil contained and allowing the shoots to grow up and towards the light. Yeah, Tony.
So when you, in your system where you were misting the roots,
Uh-huh.
the roots to me seem like they would do like hair. They would sort of go up in that because there's nothing holding them in. Do they try to do that?
Yeah. So the question was, in the system where the roots were being misted aeroponically, do the roots kind of grow everywhere? And I think the answer is, I think they just kind of occupied their volume that they had. So they were kind of contained. There was a membrane between the root shoot interface, you could say. So I think they really only were allowed, you could say, to grow within this volume. But more generally speaking, if roots are grown in darkness and there's no moisture gradient to kind of guide them one way or another, they will just grow in random squiggly lines. They won't grow straight down like they may on Earth, because roots are trying to grow with gravity on Earth, but in a lack of gravity, they just kind of grow here, there, and everywhere. Yeah.
Do you regulate the root growth with gases?
Can we regulate the root growth with gases? You probably could. I don't know.
Would they grow more if you had higher oxygen concentration below?
Uh-huh.
They grow into that.
Yeah, so if we had higher oxygen concentration below the roots, would they grow into that? I don't know if anybody's tried that. They probably would, or even if you used, like, if you injected some phytohormones, you could very well promote better or more vigorous rooting. But that sort of research, to my knowledge, hasn't been conducted. It's a great question, though. Great idea. All right. So gravity is an issue. So the plants kind of figure it out through using light as a sense to guide them to grow towards it. Another problem that a lack of gravity provides is that there's no buoyancy-driven convection. So warm air doesn't rise, cold air doesn't fall. There's no natural mixing of the air. So if I were to stay in one place on the ISS and there's no air movement, after maybe 15 or 20 minutes, there could very well be a very strong bubble of CO2 that forms around me. And I could, hypothetically, potentially, suffocate. So for that reason, crew always have a monitor that's kind of like on their collar, or maybe where this microphone is, to measure airflow around their bodies to ensure that there is always air moving across their bodies, so they don't sit and form a bubble of CO2 around their bodies. And the same is true of plants. So if we don't provide fans or air movement across the plant leaf surfaces, they can develop like a larger boundary layer, which could increase the humidity, but also can create higher concentrations of oxygen around the leaves. And I don't think I've seen this actually happen, but it is hypothetical. If you have a ton of humidity that's around a plant leaf surface, you could get cases of edema or various different physiological responses that these plants are trying to adapt and grow through, but they could really struggle. So we always have fans that are blowing air across the leaf surfaces. And this is kind of a cool way of representing that. So this flame, this is kind of like a matchstick. This flame on the right is in microgravity. So it just kind of like forms a ball around that flame surface. And in true gravity, we know that you light a match and the fire orients itself up. But on ISS or microgravity, it just kind of forms a big ball. Another issue, which I've touched on a little bit, is light. So conventionally, we could just use the sunlight to grow plants, but not really up there because they only have one window, and the crew likes to use that window because you get the best view in the world, right? So we don't put plants in that window, so we have to bring and provide our own light, and that's typically done through LED lights. This is, again, the plant habitat that we typically work with. We have a few different light LED populations that we use. So we have blue, white, red, green, and I think there's a low intensity UV light actually that we use also. So we can pre-program this light bank to provide whatever spectrum or whatever color or intensity that we need for the given circumstances. So we use light to obviously provide energy, but also to orient the plants. I'm going to try and play a video here, and I don't know if it's going to work or not. Let's see.
Yes. If you get a cloth dripping wet without...
Can you guys hear this or not? Yeah? Okay. Maybe it's picking up on my mic. I'll just...
gravity, and you wring it out, what's going to happen? What will happen to a wrung out cloth? So, and I had to use equipment that was here on board the space station. We may have the coolest washcloths ever here on the space station. I'm going to show you. Here's one of our washcloths. And it's compacted. It's put down into this little tiny hockey puck.
He's Canadian.
So that it saves space. But when you open up a hockey puck, And you pull out your washcloth. This is the one I'm going to use for the experiment today. And so when you open up your hockey puck and turn it into a washcloth, it was compressed in a great big vise somewhere. Okay, so here's my washcloth, like a magic trick. And now I'm going to get this soaking wet, and then we're going to see what'll happen when we wring it out. Meredith and Kendra suggested that I dip this in a bag, but bags don't hold water in space, so instead I filled a water bag. This has drinking water in it. And I'm going to squirt a bunch of water into this washcloth. Okay, so here's a soaking wet washcloth. Get the microphone so you can hear me while I'm talking. And now let's start wringing it out. It's really wet. It's becoming a tube of water. The water is all over my hands. In fact, it wrings out of the cloth into my hands. And if I let go of the cloth carefully, the water sort of has it stick to my hand. Okay, so the experiment worked beautifully. And the answer to the question is the water squeezes out of the cloth. And then because of the surface tension of the water, it actually runs along the surface of the cloth and then up into my hand, almost like you had Jell-O on your hands or gel on your hand, and it'll just stay there. Wonderful moisturizer on my hands. And the cloth doesn't really unravel itself. It just stays there floating like a dog's chew toy, soaking wet. Great experiment, worked perfectly. Meredith and Kendra, congratulations. Great idea.
Pretty cool, huh? Okay, so fluid behavior is a real challenge. And as you saw, like that water, as he wrung it out, it kind of just like clung to his hands. And he said like as if it were like a gelatin or something like that. So if you can imagine, think about his fingers and like compare that to like plant roots. I mean, if you put plant roots directly into just a pot full of water or a bag full of water, those roots are just going to be entirely surrounded by that water, and there's no aeration. They could essentially, there'd be a major lack of oxygen or available air. There's no gas exchange. So to combat that, we use Arslite, which I was talking about earlier. It's just stuff that they use on baseball diamond running paths. So Arslite has some really great properties in that it has good air holding capacity. It's like a crushed sintered clay. So it has a good air holding capacity, as well as an ability to hold a lot of salts, because clay has a good cation exchange capacity. It can hold a lot of salts, a lot of whatever you put into it. So we use Arslite, and we typically embed that Arslite with like a slow release fertilizer, which you're probably all familiar with. We'll just kind of evenly disperse it throughout the rooting area. And then we'll eventually, this is just kind of a snapshot of one of those root trays, and this is partially filled. And you can kind of see there's these gray tubes here. And these gray tubes are actually porous tubes. And we use those to push or pull water into the root environment. So there's some pressure sensors that are attached to the opposite end of these tubes, and based on the pressure readings that we're receiving on ISS when this is all sealed up and there's plants growing in it, based on the pressure readings, we can kind of tell if there is an abundance or a lack of water. So if you think of it like a vacuum, or maybe like a water balloon. If a water balloon's really full, there's a lot of pressure on that balloon. If it's kind of empty, and there's not very much pressure, and maybe there's actually negative pressure, as it could pull more water in or accept more water. So we use pressure to determine how saturated is the root environment within this single system. We also, additionally, we have some soil moisture sensors here. So we kind of have two senses, you could say, for soil moisture within a given quadrant. Yes, Tony?
So on Earth, if you had the roots too saturated, you would have diseases come in and kill off the roots.
Mm-hmm.
Do you have diseases on the ISS, either plant diseases or human disease, or is it sterile?
Sure, yeah. So the question is, on Earth, if the roots are fully saturated, there could be an increase in diseases, some root rots or whatnot, and are there plant and or human pathogens on the ISS? So we do our best to keep everything as clean as possible. So we actually put this arsenite into an autoclave and we try and sterilize it, and we pack everything in like clean cabinets. So we try and be as clean as possible with our plant experiments, and even like the plant seeds themselves, we surface sterilize them. Sometimes they're entirely sterilized, which means there's no germination. And that's happened quite a few times, actually. But they try and surface sterilize the seeds so they're clean, and hopefully there's no pathogens that are brought, at least on the surface of the plant seeds or in the media. Now, there are like endophytic pathogens that are within the plant seeds that hitch a ride and they make it up there. So there are some plant diseases that have popped up. I know there's downy mildew that was seen on zinnias. We don't see a lot of root rot, but not to say that it has never happened, but with this sort of system, we don't see a ton of root rot, and typically the roots are actually pretty happy because we don't overfill it, and also this arsenite just has great air exchange capacity. So we don't see a ton. I'm sure it happens, but surprisingly, we don't see a lot of plant diseases, but they are here and there. Yeah. So that's one way to kind of water plants, and that's a more active method. This is something that we co-developed with Tupperware, and essentially it's like a piece of Tupperware, but it has this cone thing in the middle. And through that cone, again, we have arsenite, and we have fins that are pointing out of the bottom of that cone. And this bottom part is kind of like a great big water reservoir. So we can have the crew fill up this water reservoir, and the plant can slowly drink from that reservoir through these fins, which absorb water, pull water into this column. And as there would be a plant here. A plant would grow. This cap wouldn't be here also, but the plant would grow out of this top here and kind of pull water on an as-needed, on-demand basis, you could say, through this reservoir. This is kind of one method that we wanted to use to kind of circumvent or save on crew time. Instead of having to have the crew come and water the plants every single day, like in other systems, you could add water to this once every 5 days maybe. So this is kind of a proof of concept, and it could work. I'll try and speed things up a little bit. One more challenge that we have is propagation. To my knowledge, there's been no asexual plant propagation that's occurred in space whatsoever. I'd love to change that, write a proposal, and take some cuttings or make some cuttings on ISS and try and root them and grow them out. And I think maybe the aeroponic system could be an interesting way to try and do that, but nobody's done that thus far. But there have been some multi-generational seed studies. So here we have some Arabidopsis, which is the model plant in the plant world, growing within this growth chamber, and these are probably about 3-week-old plants. As soon as they started to form flower stalks, we put bags over them, and we captured these flower stalks within these bags. And once these flower stalks grew near the top of the bags, we zipped them shut. And after these plants flowered and formed seeds, we then snipped them from the bottom and allowed them to kind of dry out. And with that, we successfully, captured these seeds in 4 separate bags, 1 bag per each quadrant. We then brought those bags back to Earth and then replanted another root tray with seeds that were sown from space, half of which were from space, the other half were from a ground control. and then we did the whole thing over again. And so we effectively created 2 generations of space-grown Arabidopsis, which could then be compared to 2 ground generations of Arabidopsis. So you can kind of rule out the chamber effects. You can that was a variable, but by growing 2 generations, some of which were in space for both generations, the other half was just in space for 1 generation. You can kind of rule out the effect that the growth chamber may have had on the plants themselves. The second generation of seeds from space grew and germinated just fine. There were certainly differences. This is more in like the epigenetic level, inherited genes and traits that were formed or activated because of their unique environment. There were traits that were different, but the plants still grew and flowered just fine. And then on the right here, we have some pea plants. And these peas were grown by the Russians in the early 2000s. And they grew 6 generations back to back to back to back to back of pea plants. Peas are quite large. They could harvest them pretty easily. So they would harvest them, let them dry out, replant them. And they did this 6 times over. And they didn't really notice any physical differences from the plants that were grown on space to those on Earth. So it is kind of a good proof of concept that we can grow multiple generations in space. There's more work to be done there, but it is a good groundwork, and it does give us some sense that you could grow maybe up to 10 generations of peas, maybe without any consequence. We're not sure, but 5 or 6 was pretty impressive. Now we'll look to the future a little bit, and I'll try and be quick. Again, we have some artist renderings here. This guy is actually a NASA scientist named Ray Wheeler, and supposedly he inspired the character from The Martian. The author derived a lot of information from Dr. Wheeler to help write his character. So we're going back to the moon, and Artemis 3 will actually put some science experiments on the surface of the moon, and one of which is this chamber called the LEAF, and that's another fancy acronym. I'll let you read that. But they're going to grow a few different plant species on the lunar surface, one of which is duckweed, another of which is our classic Arabidopsis, which we use for a lot of different space flights. And then we have Brassica rapa, which is like a field mustard. So they're going to grow these plants on the lunar surface, again, see how they respond. Again, we're using Arabidopsis because it's a very well characterized, it's a well-known plant that they can then try and derive, how did these plants differ in their growth and in their genomics? How do they differ from Earth-grown to ISS-grown to lunar-grown plants? And what are some of the effects that the moon had on these plants? So that is supposed to happen in 2027. We'll see if that holds, but that's the current thought, and hopefully funding makes it there. Something that nobody's ever done before is actually grown plants directly in lunar soil. So regolith is a fancy word for soil. Regolith is any soil that's not of Earth. So there's lunar regolith, there's Martian regolith. And these researchers from the University of Florida actually used lunar regolith that was collected during the Apollo missions. So Apollo 11, 12, and 17. And this group here on the left is actually a regolith simulant. So something that's designed to be regolith-like, that we can use to try and emulate what the actual real deal is like. But the study was the first to actually grow plants directly in lunar soil, and they grew. Some struggled, but others grew fairly well. And this is just kind of a proof of concept that if we had to, we could hypothetically take lunar regolith, sift it probably to a uniform size, and plant some seeds, water it, and maybe you get a crop yield out of it. Question?
Fertilized at all? Or?
I don't think that they were. Yeah, I don't think they were. The question was, were they fertilized? And I don't believe that they were. That's a great question, though. So these, again, were Arabidopsis. They're grown for about 20 days, and they grew fairly well. So it's possible we could use lunar soil to grow plants into, or more likely, we'll probably use a combination of hydroponic systems, which are already very well understood and used here on Earth, and we have the gravity of the moon to help us out with that. Or we could use a combination of some regolith mixed in, maybe with some arsenite that has better properties for at least aeration. Because as you can see, these are pretty wet and they're pretty saturated. So we'll just quickly go over a few technologies that have been influenced and helped in their development by Astrobotany. So LED lights were actually first used on plants by NASA-funded research back in like the late 80s, early 90s. There's a group out of Wisconsin that had a small business grant from NASA, and they developed LEDs that were used for plant growth. And since then, we all know LEDs have just absolutely taken off. So that has been one major benefit, not just to plant growth or plant producers, but all of us really. We use LEDs everywhere in our life. Hyperspectral imaging is also something that is kind of on the horizon that's being developed. So it's a type of imaging that uses not only the visible spectrum, but a much broader spectrum. And you can actually image plants under different wavelengths, and you can actually see if plants are maybe drought stressed or stressed for fertilizer or nutrients. And you can see that through hyperspectral imaging before we can see it visually. So in a way, it's kind of a preemptive tool to try and diagnose your plants before they actually become truly symptomatic. So this is really commonly used in like satellites. They'll fly over crop fields, take pictures, and they can see where plants are drought stressed, or maybe there's a toxicity of a certain nutrient or a lack of a certain nutrient. They can then use that to inform the farmers to fertilize their fields, through spot treatments instead of just one broad spectrum treatment. They can just treat, these 3 acres for magnesium or whatever it may be. Hydroponic, aeroponic systems are extremely popular, more so hydroponics, but that's something that NASA's been working with for, 30 or 40 years now. And then there's also some space breeding that has been conducted. So again, sending seeds up to the space environment, letting them cook in that radiation and bringing them back. And actually China has been doing a lot of research with this, and they've actually developed some crops that they now grow commercially out of seeds that have been space radiated. And this radiation kind of just knocks out random genes. You don't know what you're going to get. You bring them back, germinate them, see what happens, see if there's any interesting traits. If there are, you take that plant and use it for further breeding and try and bring that trait into a crop that is commercially grown. So that's all I have. Thank you so much for your time. I appreciate your patience. Are there any questions or comments?
What's the pH of regolith soil on the moon and the nutrient load, and does it vary from sample to sample?
Yeah, so the pH of the lunar regolith. I'm not sure what the pH is. It's probably in that paper. I just haven't read it, and I've read the paper. I just don't remember it. Okay. And does it vary from different sites? I know that they landed on different sites on every single Apollo mission, and there are differences. So there's a lot of like heavy metals and aluminum and silica that's in these lunar soils. And that was something that these plants did respond to, was like all these heavy metals and silicas. And even the soil itself, it's not near as ground down, you could say, or eroded as our soil is here. So it's a lot more sharp and jagged. And that apparently did influence the plant rooting. But I'm not sure on the pH. It's a good question.
On the seed studies, the seed germination studies, are they growing them to full-size plants? And in terms of crops, are they doing nutritional analysis? That's being done at Duke University as they, try to breed better nutritional plants.
Yeah. So are you asking about seeds that have been exposed to like the space environment? Yeah. So commonly they'll look for any like interesting like morphological traits. Then I think if it gets to a point where there is this really interesting morphological trait, like maybe there's larger fruits or the leaves are thicker, if we're talking about some sort of a like a lettuce or a leafy green, I think at that point they would then look into the nutritional aspect and look for any anomalies there. But it's really research dependent and just dependent on the group as well. And it's all based on what are their goals out of this research. Some simply want to see what's different, how they're affected. But I think those that actually want to use these plants for food crops are certainly looking into the nutritional qualities. Yeah.
I'm curious, has there been anything with woody plants and looking at lignin development? I know takes a little bit longer, but is there anything that affects them, and the gravitropism might be a little more apparent in something that's got a stem? Has that been?
That has been looked at, yeah. So I think the Canadian Space Agency, they flew some spruce seedlings. So obviously these were pre-germinated, pre-grown, and they were probably about yay tall, and they looked at lignin development in spruce. Actually, NASA tried to look at lignin development in Arabidopsis, and they flew the things and brought them back, and they came to realize that they hardly have any lignin in them at all, even here on Earth.
It's probably designed by a botanist.
Yeah, right. Not a horticulturalist. So they have looked at lignin content, and it is reduced in microgravity because there's not really a need for strong wood or sturdy stems because there's no forces acting upon them. So there's no need to build a stout trunk, you could say.
Other questions? You were answering most of mine. I had this running list of questions, and you kept answering them. So
good.
All right. Well, thank you very much. That was fascinating.
Good. Thank you.